acid cycle. So let's remind ourselves kind of what we did yesterday. We ended with this slide right here, where we talked about reduced coenzymes, oxidized and reduced, or why coenzymes are useful for oxidation reactions, redox reactions. So I want to remind you of this little figure. When enzymes cycle between the reduced and oxidized forms, that's important because it's kind of the fuel, but what's most important is the fact that that's allowing these substrates to be oxidized or reduced. That's what's going to allow these pathways to happen. Okay, so that's what you're going to see a whole lot in the citric acid cycle in just a minute. But it's because these coenzymes are there acting as oxidizing or reducing agents. Okay, now remember, in the NAD plus form or NADP plus form, these are oxidizing agents. Okay, so they are going to be reduced and they are going to oxidize their substrate. So that's why we call them oxidizing agents, because they are causing something to be oxidized. They themselves, though, are reduced. Okay, so that wording can kind of get confusing. We think sometimes people think, oh, NAD is an oxidizing agent, so it's oxidized. No, it's causing something else to be oxidized. Itself, though, is reduced. Okay, so keep that in mind. NADH is a reducing agent. All right. Okay, citric acid cycle. So we're going to start. There's 8 steps to the citric acid cycle. I'm going to walk through it, and then I'm going to kind of tell you what I want you to know from it. Okay, here's what I don't want you to know from it. I don't need you to memorize all the like structures of all the intermediates. Okay, it would not be a bad idea to know the names of the intermediates. Okay, I am going to want you to know what kind of enzyme, from looking at the reactant and then the next product, what kind of enzyme would catalyze that, and we're going to walk through that today and write them all down. Okay. But enzymes are connected to this, and we've spent a whole chapter on enzymes, so that's important to me. And then I want you to know where the coenzymes are involved, okay? Where we produce the reduced coenzymes in each step, okay? Or what steps do that, I should say. All right, we're going to start right up here. I want you to draw a molecule of acetyl-CoA, all right? We drew that yesterday. Write small today. Robin, write small, okay? Write as small as you can. We got CH.
Write it and shrink it.
Okay, that's right. You can. That's the nice thing. You can shrink it on that. And we got S, and then we got CoA. Okay, that is acetyl-CoA. What is the acetyl part? It's this part right here. Actually, I'm going to highlight it in blue because that will match our picture. This is the acetyl group. Okay, so the very first step in the citric acid cycle is the transfer of the acetyl group to oxaloacetate. Oxaloacetate is the end product in the citric acid cycle, which is what makes it a closed loop. Okay, the final product is actually the first reactant in the whole product, okay, or in the whole cycle. So we're going to call this, let me go to black. We're going to call this step 1. And I'm going to draw an arrow from oxaloacetate to citrate. Citrate is the first product. Oxaloacetate is the final product, but it's the first reactant, if that makes sense. And so this acetyl group right here, this portion, is going to come in and bind with oxaloacetate, and that's the blue part highlighted on top, those 2 new carbons. All right, or sorry, those 2 new carbons, and then one of them is a CO2, okay? A carboxylate anion portion be it. Okay, so these 2 come together, and that's what gives us citrate. Citrate is the first first product in citric acid cycle. Now, what do you guys think? What kind of enzyme would catalyze this step, the transfer of an acetyl group? That's step 1. We're transferred. That's, if you wanted to describe step 1, what kind of enzyme would do that? Do you guys remember? What enzyme brings 2 things together and binds them together? Do y'all have your table?
Is it lyase?
Is it a lyase? Did you say lyase or ligase?
I said lyase, but it should have been ligase.
It should have been ligase. Look right down here. So it's technically a ligase. If you're looking at subclass, it's a synthetase. Okay, catalyzes the formation between 2 substrates using ATP energy. Yeah, let's just write a ligase down though, because we actually don't have ATP involved in this step. Okay. So this is going to be a ligase. All right. Going to bring these two together. Okay. All right. Next step. So that's step 1. Step 2, we're going to take citrate and make isocitrate. All right. So if you were to analyze these two structures, let me tell you, you're going to find all the same atoms. You're just going to find them connected differently. So it kind of makes sense that the name of our product in step 2 here is isocitrate. So what are we making? Isocitrate is a what to citrate? If it has all the same atoms, but they're just connected differently. I think Abby mouthed it.
Isomer.
It's an isomer. Okay, so what kind of enzyme would do this step? I think Claire said it, isomerase. Okay, this is done by an isomerase. So all that's really happening, I'll just highlight. This OH group is just kind of jumping down to the next carbon. Okay, so it's one carbon down and isocitrate, and then the hydrogen from that next less carbon jumps up to the other carbon. It kind of, one hydrogen and one OH group just kind of switch places. Okay, step 3 and 4 are two really big deals. All right, so we're going to spend a moment of time on step 3 and 4. So step 3 is the isocitrate as your reactant. Alpha ketoglutarate is called the product. Okay, now in step 3, so I'm just going to write a 3 to save space. We're actually, what's happening? We are taking, I'm going to highlight this CO2 group, and we're cleaving it off. So we produce a molecule of CO2, so that's going to come off. You don't see that connected to that third carbon down in alpha ketoglutarate because we lose it. So we produce a molecule of CO2, and we're going to use a coenzyme, our first coenzyme, to do that. So we're going to have NAD plus is going to come through. I'm going to be changing colors a lot, sorry. And we are going to make this into an NADH, okay? And we also have another H plus, so people will write it like this, NADH plus H plus. Now, NADA is the low energy, NAD plus, NADH is the high energy product. NAD plus is the low energy reactant coenzyme here, NADH is the high energy form. And that's what's going to feed into the electron transport chain. So that's why I'm writing them in two different colors, blue, low energy, red, high energy. Okay. There we go. All right, now what kind of enzyme would do this step? What's happening? If we look at this next to last carbon down, I'll highlight it right here, this one right there. Look at that carbon there, and then look at it in alpha ketoglutarate. It has one carbon oxygen bond, and then alpha ketoglutarate has got a double bond. So we oxidized isocitrate to alpha ketoglutarate. So I'm actually going to write that step 3 is an oxidation. And NAD plus is reduced. So the substrate isocitrate is oxidized, NAD is reduced to NADH. And we're losing these two hydrogens. I'll just put an X over. We're ripping those off, and then we have a double bond to the oxygen. You'll see that? So what kind of enzyme pulls hydrogens off? You got to look at the table, that might help. What kind of enzyme pulls hydrogen? This is a redox reaction, so it probably might be helpful to go look at oxidoreductases. Those facilitate redox reactions. Now, if you're looking at this, is it an oxidase, a reductase, or a dehydrogenase? What do y'all think?
Is it a dehydrogenase?
Yeah, because it requires a coenzyme specifically, and these steps require a coenzyme. So this would be a dehydrogenase. So I'm just going to write that on here, the kind of enzyme facilitating this. I'm going to write this in a different color. Do it in pink. I'll underline this in pink. I should have wrote these in pink. Okay, so the enzymes will be in pink from now on. Okay, so there's a lot going on in step 3. So step 3 is the first step in the citric acid cycle that requires a coenzyme. And we produce the reduced coenzyme form NADH, all right? And it's high energy, and that's gonna feed into the electron transport chain. We'll see in a little bit. All right, let's keep going now. Step 4, it's kind of nice because it's identical to step 3 as far as the same things involved. Okay, now different substrate, different product. Alpha ketoglutarate is the substrate this time. Succinyl-CoA is the product. But step 4 is identical in the sense that we are going to lose this CO2 right here at the bottom. That gets cleaved off. So we produce CO2 just like we did in step 3. This is an oxidation. And we need a coenzyme to do that. I'll go ahead and draw my arrow since it's in black. So we're going to start with NAD plus is the coenzyme again. And we make the high energy product NADH once it's been reduced, plus H plus. So guess what enzyme does this? Same one. Yeah. Oh, I'm running out of space. Should say dehydrogenase. My H and my Y look like they're one letter. Dehydrogenase. Okay, so step 3 and 4 are identical. You produce a CO2 molecule. So far we've made 2 CO2 molecules, carbon dioxide, and we've reduced the coenzyme NAD plus. We've reduced it 2 times in a row using a dehydrogenase enzyme. Okay. So that was step 4. Now to step 5. Step 5 is the one step I'm not going to mention what enzymes used here because it's kind of confusing. It's a synthetase, but I'm not going to go into why. So I don't really care about why if you know what enzyme does this step, but I do care about the products of this step. What's happening if you look between succinyl-CoA and succinate? What is the difference? The difference is we are missing this green box down here. We are missing the coenzyme. So coenzyme gets attached to alpha ketoglutarate, and you have this succinyl-CoA is the product of step 4, but then that gets cleaved right back off in step 5. So this is step 5, and we're going to chop off the coenzyme that we just put on our molecule. But in doing that, we are going to phosphorylate a molecule of GDP. Now, y'all are probably like, what in the world is GDP? GDP, I mentioned this yesterday, but you probably don't remember because I just mentioned it, is like the cousin to ADP, okay? Very similar molecule, just instead of adenosine, it's a different base, guanine. So we're going to phosphorylate it and make a molecule of GTP, and GTP is immediately going to be made into ATP. Okay, so this step is important. Actually, I'm going to box it in. I'm going to highlight it because that's really important. This is the one step in the citric acid cycle where we make a molecule of ATP. Okay. Now, out of 8 whole steps, if we only make 1 molecule of ATP, that's not enough to produce all the energy that our body needs. So that's why we've got to make ATP elsewhere later on the electron transport chain. But we do actually get 1 molecule of ATP out of the citric acid cycle. And it happens in step 5. So that's what I want you to know about step 5. Okay. The next step, step 6. So we're on the home stretch now. Step 6, if we look at the difference here, we are going to form a double bond between that carbon and that carbon, these middle 2 carbons. If you look at succinate to fumarate, succinate is your reactant, fumarate's the product, we're going to make a double bond. So to do that, we have to pull off some hydrogens to make that double bond. So guess what? This is another step where we're going to reduce a coenzyme. It just happens that the coenzyme this time is not NAD, it's going to be FAD. And we're going to make FADH2. Now, we don't write the plus H plus. Both hydrogens actually go on to FAD. Okay? Make FADH2. So guess what? The type of enzyme that does this is identical to what it was in the other steps with coenzymes. It's a dehydrogenase. This is step 6. We don't make a molecule of CO2 in this step. We're just pulling those hydrogens off and making a double bond. Okay. All right, 2 more steps and then we're done. Step 7 if you look at the difference between fumarate and malate, I'll highlight it for you. We have a double bond in fumarate, but we break that double bond and we add a molecule of water. So we're gonna literally just H2O is gonna be added in right here. So this is a different enzyme that's involved here. What kind of enzyme would catalyze the addition of a water molecule? No coenzymes are needed. Let's go look at our table. What kind of enzyme? This is chapter 19, the table of enzymes. What kind of enzyme would catalyze the addition of water? Anybody see one?
Is it hydrotasis?
Maggie, good job. It's a hydratase. Okay, right here, catalyzes the addition of H2O. And we're not doing any kind of hydrolysis here. We're not breaking anything apart. Okay, so we don't need hydratase is exactly it. So go write that in our pink color. This is a hydratase enzyme. Okay, and that was, I'll label this step 7. All right, one more step and then we're done. Malate to oxaloacetate. So we're gonna remake the final product of citric acid cycle, which is the first reactant. We're gonna remake our starting material, oxaloacetate. And all we're gonna do, if you look at malate and oxaloacetate, what's the difference? Well, the second carbon down in malate has an OH group and a hydrogen bonded to it. In oxaloacetate, it's double bonded to an oxygen. So we are going to take off this hydrogen and that hydrogen. We're going to take off 2 hydrogens. So this is a common thing we've seen 3 times now. When we take off 2 hydrogens, we're going to use the coenzyme NAD one more time to do that. So NAD plus is going to make NAD. We're going to reduce one more coenzyme. This is the last time. And this is step 8. And same enzyme, a dehydrogenase. Pulling off 2 hydrogens so that you end up with this carbon double bonded to the oxygen. This is an oxidation reaction. Or a redox reaction, I should say. Okay, and that is it. That is the citric acid cycle. That's a lot. Okay. So I want to pull out, again, I don't want you to memorize everything on this slide. That's not what I'm asking for. But let's go to the next slide and kind of summarize the things that we think are most important to know here. So what is the net results of the citric acid cycle? What are the most important things to know? Number 1, the reduction of coenzymes. How many specifically? 4 total. Three, we get 3 NADH, and we produce 1 FADH2. So we reduce 4 coenzymes, we end up with 3 NADH, 1 FADH2. And those are going to be
NAD plus, would that not be a coenzyme?
NAD plus is the coenzyme that we reduce. I'm just writing what we make. These are technically what is produced.
Okay, thank you.
By the reduction of the coenzymes. The ones we reduce, we're reducing NAD plus and FAD. Those are the ones we're reducing. When we do that, we produce NADH and FADH2. These are going to be important for what we talk about next. They're going to feed into the electron transport chain. Okay, the second thing that's important, that's kind of one of the net results of the citric acid cycle, is we have converted the acetyl group. Okay, you guys remember this acetyl group up here that I highlighted in blue, our very first starting material from this CoA enzyme? Ultimately, those two carbons are converted or lost as CO2. So they're converted acetyl group to 2 molecules of, it's an S, of CO2. We added 2 carbons onto oxaloacetate in the beginning, and then we lose 2 carbons as molecules of CO2 in steps 3 and steps 4. Is where we produce those 2 molecules of CO2. Last thing is we make 1 ATP. That is one of the net yields of the citric acid cycle. Okay. So what do I want you to know? I want you to know what steps produce our molecules of CO2, and from what steps do we get reduced coenzymes, our NADH and our FADH2? What step makes our ATP? And then I want you to know which enzymes, if I said, oh, here's step 6 from the citric acid cycle, succinate to fumarate, what products are made from this, and what enzyme would catalyze this reaction? I would want you to be able to tell me, oh, we reduced the coenzyme FAD to FADH2, a dehydrogenase is the enzyme that would catalyze this reaction. But you do not, if I want you to see the molecules, the intermediates, citrate, isocitrate, I will put those molecules and those compounds on the exam for you. Okay, you won't have to draw those out. Okay, questions on this? Is this too overwhelming? Is it pretty overwhelming? Or y'all feel okay about it? It's gonna need some time. You're gonna have to look over it this weekend.
Would you give us like a whole citric acid cycle like this to fill in, or would you just give us like citrate to isocitrate?
Probably not a whole citric acid cycle.
Okay.
I'm not gonna make you write everything we just wrote.
Okay.
All right, any questions on the citric acid cycle? If I ask you, what is the first reactant of the citric acid cycle, what would you say?
Oxalate, right?
Close, oxaloacetate. It's a hard word to say. All these are hard words to say. Oxaloacetate, yeah. And if I ask you, what molecule is like the starting material for, what molecule feeds into the citric acid cycle? It's the starting material for the citric acid cycle. What would you say?
Would it be the acetyl?
Acetyl-CoA, this molecule right here. That is that feeds into, that is the net result of digestion. Okay, all the carbs and lipids and proteins that we eat, they're digested also that we can make acetyl-CoA so that it will feed in. So we're never short on acetyl-CoA in our body.
Wait, so what's ligase again in the first step? Is that
The ligase is the kind of enzyme that catalyzes this reaction.
okay?
It's some kind of ligase. All right, questions there? Any other questions? If not, we're about to move on to oxidative phosphorylation. I'm really excited. Okay, oxidative phosphorylation. So this is how your body makes ATP. And it's really, really cool. I think you're gonna think it's cool. So it is the synthesis, if we define oxidative phosphorylation, the synthesis of ATP using energy released into the electron transport chain. Now I want you to think about the two words, oxidative phosphorylation. So something is going to be oxidized, something's going to be phosphorylated, okay? So be looking for those things when we talk about the electron transport chain. I want to remind you that all of this is taking place in your mitochondria. So this is kind of a cartoon image of a mitochondria, not the exact same one we looked at yesterday, but you can see the outer membrane, the inner membrane space, and the matrix is the part colored in that brown or orange color, okay? So the matrix is where the citric acid cycle, you can't really see red, I bet. This is where the citric acid cycle is taking place all in the matrix, and then it's across the inner membrane into the inner membrane space is where we're going to make ATP, okay? All right, so let's define a few things. There's two major components to oxidative phosphorylation. The first component is what we call the electron transport chain, or I'm going to abbreviate it ETC. And this facilitates a series of oxidation reduction reactions that pass electrons from the reduced coenzymes that we just made. So the first blank is reduced coenzymes, ultimately to oxygen. So the reason you breathe oxygen, the reason oxygen has to go to every cell of your body, is so that that oxygen can come into your mitochondria and accept electrons, okay? That is the reason we breathe oxygen. Isn't that cool? So the final result is gonna be, we're gonna make a molecule of water, and we're gonna get a proton gradient. Okay, you guys are familiar with the word gradient now. We talked about that when we talked about the bilayer. So across the inner membrane of the mitochondria, we are going to get this proton gradient where one side is going to have a high concentration of hydrogen ions. So when I say proton, by the way, what I mean is H+ ions. Okay, I talked about that, how that hydrogen atom that has lost its electron and now become a cation, all that's in there is a proton. So that's why we just call it a proton gradient instead of saying a hydrogen ion gradient, because that's harder to say. So one side of the membrane is going to have a high concentration of hydrogen ions or protons. One side is going to have very little. And it's that separation in the concentration that's going to be used to do a lot of useful work to make our ATP. Okay, the second step, if you read about this any on the internet, like you're trying to like help teach yourself and find, there's really great videos out there, by the way, really great articles. If you just want like another summary to read over this weekend to help prepare yourself, but you might see this word chemiosmosis. So I just wanted to define it in case you come across it and you're like, what? We didn't talk. It's just the process of using energy from a proton gradient to make ATP. That's what that word means, in case you see it. Okay. You'll see this figure again. I have it on a slide down below, but this is kind of the simplified version of the citric acid cycle. And it shows you which steps give us the reduced coenzymes we talked about, which one makes the GTP. Now, the reason it says like C2, C6, C4, this is just telling you how many carbons are in each of those intermediates. So you don't have to pay attention to that. I would go through and just number this: step 1, step 2, step 3, step 4, blah, blah, blah. But you don't have to do that either. So this is just a simplified version of the citric acid cycle for you to see and to remember what steps give us what. Okay, so I'm going to tell you kind of summary here. We're going to go through the electron transport chain in 3 layers. And every layer is going to add something. So it's going to get kind of more and more complex because there is a whole lot going on in this stuff. So the first layer is going to be that we are going to follow the path of electrons through the electron transport chain. We're just going to see how they move through the proteins and through the membrane. The second step is we're going to look at how the movement of electrons transfers energy and releases energy. Okay, and then how that energy is used to pump protons. Remember, protons are hydrogen ions. The last step is we're going to talk about how the gradient created from pumping those protons drives the synthesis of ATP. Okay, so step, or I should say layer 1 is, let's go to this slide right here. Okay, so you have this beautiful picture here, cartoon picture here. I have a color-coded key down here for you because there's a lot of different colors. So this is a membrane. This is the inner membrane of your mitochondria. So orient yourself real quick. We're going to say the top is the inner membrane space and the bottom, beneath that bottom line, is the matrix. Okay. So in the inner membrane, we have these proteins, we call them protein complexes sitting in the membrane. And we talked about integral proteins, right? They span the whole bilayer and they transport stuff. They act as channels or pumps. Things can pass through them, okay? These proteins are pumps. They're gonna use to pump, they're gonna be used to pump hydrogen ions. So we have them labeled 1, 2, 3, and 4. You can see the Roman numerals in the bottom corner of each one. And these are stationary, they don't move, but they transport electrons and pump protons. Now the one that's different is number 2, complex number 2. Complex number 2 is different because it doesn't span the whole bilayer, right? We call it a peripheral protein. You don't have to write that down, but it's not an integral protein. So it's not used as a pump. The only ones that are actual pumps are going to be 1, 3, and 4. So complex number 2 is not a pump, but it is used to transport electrons. All right. The yellow things that you see, one says CoQ and one says CYTC. Okay, these are mobile electron carriers. They are just going to shuttle electrons between the protein complexes, between 1 and 3 and 3 and 4. Okay. And coenzyme Q is CoQ, and cytochrome C is that second one up top. Then the green thing that you see is ATP synthase. This is a huge protein with 2 gigantic parts, and it looks really complicated, and it is. But that's ultimately going to use the energy from the proton gradient, and that's where ATP is going to be made. Okay, so it's called ATP synthase is the name of it. All right, so let's actually follow the electrons. So what happens is when we run the citric acid cycle, we know we produce NADH and FADH2. And this is all in the matrix. So those coenzymes immediately go to the membrane, and NADH is going to be oxidized. FADH2 is going to be oxidized at complex 1 and 2. NADH always goes to complex 1. FADH2 always goes to complex 2. And these are going to be oxidized. You can write oxidized right there, back to their original forms, NAD plus and FAD. Okay. So the reduced coenzymes go and they're oxidized back into their oxidized forms. And then these just cycle right back to the citric acid cycle to be reduced again. And that's all they do. They just shuttle back and forth between the citric acid cycle and the membrane. They pick up electrons, they carry them to the complexes, they drop the electrons off. Okay. So these electrons, that's what the arrows are, the movement of electrons. So these electrons are going to be given to complex 1 and to complex 2. Actually, I'm going to write these in black. I think you can see that better. Complex 1 and complex 2 are going to now receive the electrons from our reduced coenzymes. Okay. They're immediately going to be given to the first electron mobile carrier. At first, this mobile carrier is sitting in its oxidized form, but when it picks up the electrons, it's going to be now reduced. Remember, the gain of electrons, we call that reduction. The loss of electrons, we call that oxidation. So a bunch of redox reactions. This electron transport chain is just a series of redox reactions. So coenzyme Q is just going to shuttle those electrons over to complex 3. And in doing so, it's going to be oxidized. And then those electrons are going to go from complex 3 over to the next mobile electron carrier, cytochrome C. Now, cytochrome C is a metalloprotein. Okay, you guys remember what a metalloprotein is?
It would have metals, wouldn't it?
It has a what, Abby?
Like metal groups.
It has an ion in it, a metallic ion. Okay, and ion or an atom of iron, specifically an iron with a +3 charge. Some people will write it Fe with a Roman numeral 3. Okay, but an atom of iron +3 is sitting there waiting to receive these electrons. I mean, look at all the positive charge on iron. It's happy to receive electrons. There's a lot of positive charge. We know positive and negative attract. So those electrons are super attracted to that iron with the +3 charge. When it receives the iron +3, it becomes iron +2. It's reduced. Okay, you can think of the charge going down. When we receive an electron, our charge goes down by 1 because we picked up something negative and we're reduced. And then all it does, it's a mobile electron carrier, so it's going to shuttle that electron right over here to complex 4. The electron is going to pass through complex 4 and ultimately be received by the last receptor in the electron transport chain, and that is oxygen. Oxygen is going to receive those electrons, combine with 2 cations, hydrogen cations, and make a molecule of water. Now, this might look a little confusing to you. You might not have seen something like this before. We have a 1/2 in front of this oxygen atom, just to indicate that it's 1 atom of oxygen. Okay, we breathe in a molecule of oxygen, but that oxygen is then split in half into 2 individual oxygen atoms, and both get 2 hydrogen ions, and we make 2 molecules of water. But to balance the reaction, we just write it like this. Okay. So all of this is going to happen 2 times, and it's going to give us 2 molecules of water. Okay, so that's the first layer. That's the movement of electrons through the chain. Okay, questions so far?
Are you going to tell us what we exactly need to memorize and know about these layers? Like what you would want us to memorize and know?
If I give you a blank one, could you label it?
The whole thing? You got me.
Hang on, let me finish, Robin, and then we'll talk about that, okay? Let's go through the next 2 layers.
Oh, wait, I have a real quick question. I know
Yes,
you
Claire.
probably already explained it, but why does it go through it twice?
Why? Well, the electrons pass through because we have 2 coenzymes. We have FAD giving electrons, and we have NAD giving electrons. So they're both sending electrons. So multiple electrons are passing through.
Okay.
And I just want you to know, for each molecule of oxygen, O2, this breaks apart into O, and this breaks apart into O, and those both make H2O.
Okay. Okay, that makes sense.
Does that make sense?
Yes.
We
Thank you.
split that oxygen molecule in half, and both of them make a water.
Okay.
Yeah.
Thanks.
Any other questions so far? Okay. I want you to know the difference between the complexes, which ones are integral and which one does not pump protons. All right, now let's talk about how energy is involved here and how the movement of electrons through these complexes releases energy. So for the electron transport chain to happen in a sequence, there's a lot of different steps, right? And they have to go in order. So this is a sequence of reactions, redox reactions. So for the sequence to continuously happen, every step has to be favorable. Now you guys remember delta G? What kind of delta G indicates a favorable reaction?
A negative.
Negative delta G.
Good, okay. So I'm gonna write this up here in the top corner. Every reaction here, every transfer of electrons has to have a negative delta G, and it does. Every reaction in the electron transport chain has a negative delta G. Every reaction has to be favorable for this to happen in order. So what that means is that you have to start out with something very, 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 very high energy so that you can continuously, with each step, give off a little bit of energy, little bit of energy, little bit of energy, and kind of climb down the ladder to lower energy products, okay? So you can think of it kind of like this waterfall here. I'm gonna draw on this picture, and I was playing around with this yesterday. Tell me if you can't see the color here. I felt that yellow was the best color, but if it's hard to read, just let me know. Think about it as like steps of a waterfall, and I'm gonna draw a y-axis on our picture here. So our y-axis is gonna be energy, okay? Delta G, free energy, okay? So at the very, very top of the waterfall, I mean, think about water, actual water in a waterfall. At the top of the waterfall, that water has the highest amount of potential energy, right? What's the difference between potential and kinetic energy? When I say the word potential energy, what do I mean?
Isn't that like stored energy?
Yeah, stored energy. The moment that water falls down the waterfall, it's moving. It has kinetic energy. Water at the very, very top water held up, okay, there's a gravitational pull on it, has a lot of potential energy. It's like water stored behind a dam. We talked about the hydroelectric dam yesterday. It's a lot of stored energy. So that would be up at the very, very top. I'll write in black first, but that's going to be our reduced coenzymes. They have the highest amount of energy. They're the highest energy carriers. When they pass electrons, I'll go back to yellow. When they pass electrons off, every step releases energy. So that means a little bit lower in energy would be complexes 1 and 2, because those are both in tandem the first step that we pass electrons to. They're a little bit lower in energy. Those pass electrons to complex 3. A little bit lower in energy. We release energy. Okay, so with every step, we're going to release energy. Can y'all see the yellow?
Yes.
And then we're going to drop to the lowest, complex 4, and then to water. Okay, and every step is going to release energy. So as you can imagine it, down this waterfall, it's the same idea through the electron transport chain. So I'm literally going to write the same things over here. Passing electrons through complex 1 releases energy. And I'm going to try to copy and paste this because I don't want to write it 6 times. Same idea here. In complex, no. I'm not very good at this yet. Copy, paste. And then paste. Okay, so write this on complex 1, 3, and 4. The movement of electrons, this looks a little deceptive, the arrow, I'm going to write it like this, releases energy, releases energy. Okay. All right, now the energy, fill in this blank down here, released from the movement of electrons through these redox reactions, powers the transfer of hydrogen ions from the matrix to the intermembrane space, creating a proton gradient. All right, so let's actually draw that. Literally, what we're going to do here is we have hydrogen ions sitting in the matrix. And as the electrons are passed through these protein complexes, they're just going to be pumped across into the intermembrane space. I actually wanted to do this with a very thick line. Maybe that'll show up better. Okay, just like this. Back to here. Okay, and that's the end of layer 2. The movement of electrons is energetically favorable, releases energy, and then we pump the protein, or I need to write H pluses over here now. Pumps these hydrogen ions across the membrane against the gradient. Okay, so this is, what kind of transport is this? Active or passive? Definitely active, right? Requires all kinds of energy. So here's our color code thing again, if you need that key. Now we have the added, these arrows still represent everything you see. The gray arrows still represent the movement of electrons. Okay, layer 3, look how complicated this looks. But I just wanted to keep everything on there from the previous 2 layers. So now you see the release of energy and you see the movement of the hydrogen ions across the, don't ask me how long it took to make this slide. Okay, so now let's talk about the effects. What, Robin?
I said, I'm proud of you. Good job.
Thank you, I was proud of it too. The effects of pumping protons, okay? What does this do? Well, we know that hydrogen ions correlate to pH, or at least we should remember that, okay? I'm gonna write up here in the very corner, high amounts, it's not thin enough for me. High amounts of hydrogen ions, a high concentration of hydrogen ions correlates to a low pH. That's an acidic pH. So as we're pumping all these hydrogen ions across the membrane, one side of the membrane becomes very acidic compared to the other, which would inherently be basic. So the intermembrane space becomes acidic, low pH. The matrix becomes basic compared to the intermembrane space. Now, we're not talking a gigantic difference here, because we need our cells and all the other proteins need the pH to stay relatively stable. Remember, crazy low or crazy high pHs can unravel and denature proteins, right? So we're not talking crazy change in pH difference, but it's enough of a difference. Okay, so that's the first thing. The first effect is a pH difference. It's enough of a difference to create a potential energy difference. Okay, so a difference in the concentration of H plus creates a gradient, and that gradient has potential energy. Just like we were talking about, you guys remember, go back to that picture of the hydroelectric dam. We have now created a store of water behind this dam. When I say water, I mean H plus ions, hydrogen ions. And there's a ton of potential energy there, just like there is with any hydroelectric dam that stores a ton of water behind the dam. Okay, so the difference in pH, the difference in hydrogen ion concentration creates the second effect is a potential energy difference. All right, now we're going to use that difference. We're going to channel these hydrogen ions back across the membrane and harness their energy to make ATP, okay? Because think about it. What would happen to a hydroelectric dam if you suddenly just made the wall of the dam disappear? How much force would that water have if it just like rushed through the river or the canyon or whatever's in front of it? A huge amount of energy, right? Same idea here. If we just removed the membrane and all these hydrogen ions could flood back across, that would release a ton of energy. So we have a huge potential here. We want to harness it and use it to do something useful for our bodies. So we're going to channel the flow of hydrogen ions. We're going to channel it where we want it to go. And we want it to run through ATP synthase. Okay, that's the only way back across the membrane is through ATP synthase. And these hydrogen ions are dying to get back across. Okay, so let's look at ATP synthase for just a minute. It's this huge protein complex. It's got two portions, F0 and F1. The F0 portion is what facilitates the return of the hydrogen ions back to the matrix. Okay, the F0 facilitates the return of hydrogen ions back to the matrix. The F1 portion contains the binding site for ADP in a phosphate group. Okay, now we have two different subunits, so that's why you see the little alpha and beta here. There's actually 6 subunits here. Don't write this down. There's actually 6. You only see 3. Okay, but there's actually 6 of these oval looking subunits on the bottom of F1, the F1 subunit. And all the beta subunits are where the ADP and the phosphate group bind so that they can come together and make ATP. Now, as the hydrogen ions pass through ATP synthase, they literally cause this F1 unit to rotate. So remember we talked about the turbine in the hydroelectric dam, how the water causes it to mechanically rotate? Same thing happening here. The F1 subunit literally rotates. And this rotation is what brings these two together. That's where the catalytic activity happens. That's why it's synthase. It brings the phosphate group and the ADP together to make ATP. And this costs a lot of energy. Forming ATP costs the same amount of energy, same number-wise, as the hydrolysis of ATP. So it costs us, our cells, our body, 7.3 kcal of energy. But when we have this huge proton gradient difference, it's easy for us to supply that energy. Okay, but we have to create it first. And it takes a lot of steps, right? Why do you think it takes so many steps to build up that proton gradient when it's all released to make ATP back in one step? Okay, that's because it's a pretty big amount of energy that we're talking about. Now remember, 7.3, technically in the grand scheme of things, is an intermediate value. There are other atoms or other things that it would cost us more to make. But again, we don't want to get too costly. Okay, the body, this is already a high enough amount of energy to invest to make ATP. Okay. So now let's talk about the net yield of ATP synthesis. How many hydrogen ions have to flow through ATP synthase to make 1 molecule of ATP? So it's the transfer of 4 hydrogen ions back to the matrix that can make 1 molecule of ATP. And we start off with 1 NADH and 1 FADH2. So how many of these reduced coenzymes do we have to feed into the electron transport chain to make ATP? Okay, so electrons from 1 molecule of NADH pumps 10 hydrogen ions across the bilayer into the intermembrane space. Electrons from 1 FADH2 pumps 8 hydrogen ions. It's a little bit lower. FADH is a little bit lower energy than NADH. Into the intermembrane
Could you
space.
show us that on the picture?
What, Maggie?
Could you show us that on the picture, like where that exactly happens?
Oh, these pumps right here. So we could say NADH pumps, you could write 10. Okay, it pumps 10 hydrogen ions. FADH via complexes 1, 2, and through 3, pump FADH pump, I don't know where to write it because there's not a direct line across, but it pumps 8 hydrogen ions across the membrane. So I'll write that down by FADH2. Does that make sense, Maggie? I don't know if that helps you. So
Yeah,
from
I
one,
was looking at something different.
oh, did you want me to go up and write it somewhere else?
No, I got it now.
Okay. So our reduced coenzymes essentially pump 18 hydrogen ions across the membrane between the 2 of them. So if we could, you'll see this too. And in fact, if you read through this section in your textbook, how many ATP can we get from 1 NADH? And how many ATP can we get from 1 FADH2? Can you guys calculate that? It's a little bit of stoichiometry, if you remember that word. We have relationships here. So if we have, for every 1 NADH, we pump 10 hydrogen ions. So if we have 10 hydrogen ions, how many ATP can we get from that? You need to write something like this. If we have 4 hydrogen ions, we know that makes 1 ATP. So 10 divided by 4 is what?
Two something.
2 something. 2.5 ATP. So you'll see your textbook rounds up and it says for about 1 molecule of NADH, we get 3 ATP. They just round up. Okay, what about FADH2?
Would it be 2 ATP?
Mm-hmm. So we get 2.5, or some people say 3 ATP from a molecule of NADH. We get 2 ATP from 1 molecule of FADH2. Okay. So roughly 4 to 5 ATP for every set of coenzymes that feeds into the electron transport chain. Okay, this is the 3 layers. This is oxidative phosphorylation. So what's oxidized and what's phosphorylated? Why do we call it oxidative phosphorylation? What do y'all think? Why do we call it oxidative phosphorylation? What is oxidized and what is phosphorylated? Anybody remember? So we wrote it down on layer 1. It's the reduced coenzymes that are oxidized. And the reduction of these reduced coenzymes ultimately through a chain, a series of steps, is coupled to the phosphorylation of ATP. You guys remember how we talked about coupled reactions? These work in tandem. If you stop one, you stop the other. What's actually really impressive is the fact that there have been a lot of weight loss drugs over the years that have come onto the market that mess with our ability to make ATP. And we don't realize that until we do a lot of research on it. And then these drugs are pulled back off the market, but they're very dangerous. Okay, so people who get into like bodybuilding, and they're trying to really trim their bodies down and lose weight, and then sometimes they rely on supplements to help them do that. And you have to be very, very careful. Okay, if you overdose on those supplements, they can be fatal. It's really crazy when people mess around with that stuff, because those supplements are literally messing with our electron transport chain and oxidative phosphorylation. So I've got a few poll questions that we're going to go through. I don't know if you have both slides. Yours might be all on one slide. Sorry if it's crammed on there. I've tweaked this last night. Do you actually even have these questions? No? Okay. So screenshot this if you want. That's a lot to write down, so don't try to write all this down. But I want you to read these questions. Let's look at the first one. The first one's the easiest. Coupling reactions is one of the major metabolic strategies within oxidative phosphorylation. What is the synthesis of ATP coupled to? Oh wait, I didn't write the, do I even have the right?
No.
Oh, this is the wrong poll. This is a poll from yesterday. Don't answer this one. I didn't want that one. Did I do this one? Okay, I don't have a poll on this one, so we'll just answer this one together. I have a poll on the next two. What do y'all think? Looking at the answers here, is it coupled to the reduction of NADH and FADH2, the dissipation of the hydrogen or proton gradient, the phosphorylation of glucose, or none of the above?
Is it the reduction of NADH and FADH2?
Oh, I want to hear from some more people. What do y'all think? Anybody think something different? Nobody has any other thoughts? So we're looking for a reaction. This right here is not a reaction, okay? But this is an oxidation reaction, okay? Or sorry, you know, that might actually also be a typo. This should say the oxidation. So it should actually be none of the above. But that is totally on me, all right? NAD and FAD are reduced in the citric acid cycle, and they're oxidized in the electron transport chain. So they're not reduced when you come to they're oxidized. So the correct answer would be, nope, it's not coupled to the reduction of NADH, because NADH is not reduced, it's oxidized, and so is FAD. Does that make sense? And that's why it's called oxidative phosphorylation. So the correct answer there would be none of the above, but if it said the oxidation of NADH and FADH2, then you would pick that. Because that is what, yes, Abby.
So if that did say oxidation, does that have anything to do with the feedback, like going back to the very beginning? Or am I just overthinking this?
Going back to the very beginning, what do you mean by the very beginning? Like the first layer?
Yeah.
Yeah.
Okay.
Yeah, right here, this is the very first, if I had to number the steps of the electron transport chain, that's the very first step. The oxidation of NADH and FADH2. Step 2 would be the movement of electrons through the chain. Step 3 would be the pumping of hydrogen ions across the membrane. Step 4 would be the passage of those hydrogen ions back through ATP synthase to make ATP.
Okay, thank you.
Okay, I do have a poll question for this next one. So I'm going to read the problem first. So I don't want you to just know the electron transport chain. I also want you to know some scenarios that are described here. There might be some more questions in the textbook for practice on this, of how different things can mess with our electron. If we messed with it somehow, or we uncoupled something, or something broke down, how would that affect our electron transport chain? So that's what this problem gets at. It says cyanide acts as a poison because it inhibits complex 4 and it takes away its ability to transport electrons. How would cyanide poisoning affect the electron transport chain overall and the proton gradient across the inner mitochondrial membrane? So if we inhibit complex 4 and it can't pass electrons back across the membrane, the electron transport chain would speed up and the gradient would become stronger. The electron transport chain would stop, the gradient would decrease. Both the electron transport chain and gradient would stay the same, or it would be rerouted through complex 2 and the gradient would become weaker. So I'm going to launch this poll. Don't say your answer out loud. Just put it on the poll, A, B, C, or D. What do y'all think? All right, 2 more. Okay, great. I don't even have to talk about this. Everybody picked B, which is totally correct. You would shut off the electron transport chain. You cannot continue oxidative or the redox reactions if you stop it at one point. They all have to run in sequence. So the electron transport chain absolutely would stop. And if we stop moving electrons, we stop pumping protons. Okay. So the gradient would dissipate. Okay, another poll question. Oh, question there, Claire?
Oh, yeah, I just wanted to double check. So if any one of them, like if 1, 3, or 4, if any one of them stopped working, they would all
Yeah.
Okay.
Yep, if you inhibited any of those complexes, you would shut off the chain. Electron
Okay. Right, that makes sense. Thanks.
Okay, this next question gets talks about the drug I was actually mentioning earlier. So this drug, dinitrophenol, DNP, is a chemical that acts as an uncoupling agent, making the inner mitochondrial membrane leaky to protons. Think about that, leaky to protons. Let's look at the inner mitochondria. What it's saying is that now these hydrogen ions can just pass through on their own. They can come back across on their own. It was used until 1938 as a weight loss drug. Oh my gosh. How would DNP affect the amount of ATP produced in cellular respiration? So in addition to calling this oxidative phosphorylation, it can also be known as cellular respiration. Why? Because this is why we breathe oxygen. This is the purpose of breathing oxygen. Why do you think it's off the market now? So did I launch this? Launch the poll. A, it would increase ATP production, but also cause dangerously high body temperature. It would decrease ATP production, also cause dangerously high body temperature. It would decrease ATP production, but cause dangerously low body temperature. Or lastly, would increase ATP production, but cause dangerously low body temperature. So why are we talking about body temperature in this?
Well, I assume the faster things go, the more heat it creates.
Energy is a type. Heat is a form of energy. Should put it that way. Heat is a form of energy. So if we're releasing energy in our bodies, we're releasing heat.
I want to change my answer.
I don't, yeah, okay, everybody's voted. Okay, in the poll, we got some different results here. I'm gonna share what we got. So let's talk about this. What do you, I want to hear from y'all. What do y'all think? The most commonly picked answer is C.
I picked C, but I think that it would decrease the production. But when you said that energy like creates heat, if we don't have the energy to give off the heat, I think it'd be a high body temperature, I think.
Okay. Somebody else, somebody that actually picked B as your answer.
I picked A because I figured the more reactions it would have, the higher body temperature would be because of the more reactions.
Okay, and that we would increase ATP production in that.
Yeah.
Okay,
Yeah, I picked A as well because I figured if the hydrogen ions are able to move back and forth, that it can create as much ATP as it wanted to. That's what was my thinking behind it.
okay. Somebody who
picked-
thinks that it would decrease ATP production. Why did you pick that?
I picked that it would decrease ATP production because wouldn't it technically have to go through the ATP synthase
Stop.
to be made into ATP?
Sorry.
Robin, are you okay?
Sorry. I didn't know I was not muted. The dogs started going crazy.
Okay, so Claire said she picked that it would decrease ATP production because the hydrogen ions have to move through ATP synthase to make ATP. This was her thought. So let's go look at, I'm going to highlight a couple things on our problem, then we're going to look at the picture of the electron transfer chain. It's saying that this DNP drug is an uncoupling agent and it makes the inner mitochondrial membrane leaky to protons. And what I drew on here, as I said, now these hydrogen ions can literally pass back through the membrane without going through ATP synthase. Like they can literally just flow. They can flow back across on their own without going through ATP synthase. Okay. So to make ATP, the whole premise here is that we are channeling these hydrogen ions through the proton or through the protein ATP synthase. You have to pump them through ATP synthase to make ATP. So if they can now move back through the membrane on their own through simple diffusion and not active transport, then we can't maintain that gradient. And the whole time they're rushing back across because the gradient is going to direct them back across to the matrix, they're releasing tons of energy. Tons of energy would be released because they're all going to rush back across at once if they can. That's what they want to do. It's just we're making them go through ATP synthase so that we can use that energy to do something useful. And if the movement of just 4 hydrogen ions releases 7.3 kcal of energy, what if now 10, 20, 100, thousands of hydrogen ions can rush back across the membrane uncontrolled? Think about how much energy that's going to release. And again, if heat is a form of energy, what's that going to do to our body temperature? Dangerously high body temperatures. So we're going to stop making as much ATP. Maybe some hydrogen ions will still move through ATP synthase. But we're going to stop making as much ATP. So we're going to feel tired because we're low on energy. And our body temperature is going to shoot up. And that's exactly what was happening with DNP. Is these athletes, these bodybuilders, would die if their body temperature, it would become too fatal sometimes if they took too much of a dose of DNP. It's a very, very fine line between what could be a controlled effective dose and what could be an overdose. Okay, does that make sense? So the correct answer there was that it would decrease ATP production, but cause dangerously high body temperature. So B was the correct one there. Okay. Wow, 6 slides, and it took us almost an hour and a half. But now I think you see why. That's it for today. That is all of chapter 21. If you read through chapter 21, we didn't necessarily hit on every single section of chapter 21. But I don't discourage you from reading through the chapter at the same time or skimming through it. It's got a lot of these helpful figures, and it's got other figures that I don't even have time to put on here. If I sent you the PDF yesterday, don't worry about all these excess slides. I'm not going to be asking any questions about these. Okay. I realized I probably sent y'all that yesterday, but we're stopping here. Let me see. Close
So,
that.
I don't know if
Maggie,
you already,
yes?
could you repeat what you wanted us to know for the electron, transport chain?
Yes. I remember Robin asked me that. Okay. So I want you to be able to, I want you to know the 3 layers. There's really not a simple way to put it. This is a lot of details, I know. I want you to know how electrons, if I gave you a blank diagram, I would want you to be able to draw how electrons are going to pass through there. I want you to know what the initial reactants are, that it is NADH and FADH2, the reduced coenzymes that feed the electron transport chain. That's the oxidation of those, that I want you to know that those are high energy molecules. Okay, so the movement of electrons. I want you to be able to tell me if I ask you about cytochrome C, that it's an atom of iron that cycles from iron 3 to iron 2 as a mobile carrier to pass electrons through. Cytochrome C is an example of what? It's an example of a metalloprotein. Okay, all of these are fair questions based on what we've talked about. I want you to be able to talk me through the way energy is released at every step of the way and why that's important. That's important because all these reactions have to occur in sequence. They all have to be downhill reactions. Remember yesterday we drew that energy diagram with the downward arrows? These are all exergonic. If I ask you, is this an exergonic or an endergonic reaction? Okay, we did a little bit of a review yesterday at the beginning on delta G and what those terms mean. Negative delta G is scientifically, thermodynamically called exergonic. Positive delta G is endergonic. Okay, so all the reactions through the electron transport chain are exergonic. I want you to tell me what the final products are. What are the final products of the electron transport chain? We make something from the passage of electrons, and we make something from ATP synthase. I want you to tell me quantitatively, how many ATP do we get? What if we fed 5 NADH through the electron transport chain? How many ATP could we make? What if I told you a rate? Oh, we do 5 an hour or 500 an hour. How many ATP could we make?
That's a lot of math.
Oh, that's not hard math.
I don't like math.
Well, I can't help you there.
So
Yeah,
do
that sounds like
I
a
need
lot
a
of points.
calculator?
Do what? I would bring a calculator to the exam, not your cell phone.
Can you
If
send
we
us
did
a
500
reminder?
NADH an hour, how many
That's like
ATP
100
could we make in an hour?
right?
Well, you know that for every 1 molecule of NADH, you make 2.5 ATP. So you just do 500 times 2.5.
That's hard in my head.
That is not hard.
Simple math is hard.
If you want to climb the ladder in administration at a hospital, you're going to have to be able to do math like that at least.
I will
Do
have a
1250.
calculator.
Then you're going to have one for the exam. Bring one for the exam.
Can you send out an email the night before or something? Say, hey, bring a calculator because I didn't remember. Please.
Then you can set an alarm on your phone right now that reminds you
Okay.
on Monday morning to bring a calculator.
Yo,
Okay,
Robin,
Cameron's been trying to say something. Cameron, sorry, talk.
I was just saying that sounds like a very big question, a lot of points.
I think Cameron's trying to trick me. I think he thinks that's easy math and he wants me to make that worth a lot of points so that he gets a lot of points from easy math.
Hey, don't hate the player, hate the game.
And then the questions like what we talked about, if I manipulate the electron transport chain, like if I have some kind of drug that does something to it, how would that affect the production of ATP? How would that affect body temperature? How would that affect our reduced coenzyme? Whatever, okay? I might ask you some things like that.
Is the quiz gonna be hard?
That, I can't answer that for you. I don't think it's hard.
Oh, well, of course you don't.
Yeah, so I don't know how to answer that.
Okay, this is kind of a dumb question, but for doing math, it's like the time necessarily doesn't matter, right?
No, you would do something, you'd make this much ATP in an hour
Okay, cool.
or whatever.
So you could say like 30 minutes and it would still be
Yeah,
like,
30
okay.
And then it's one day.
So how? No questions are dumb, Claire. By the way, because this.
Yes,
one-
Claire, I agree. No questions are done.
So like we're gonna know what we need to multiply by, right? Like 2.5 or 2. Like it's gonna be like NADH or FADH2. Like you're gonna tell us.
Oh, you can't see my screen. I zoomed in on it. 1 molecule of FADH2 makes 2 ATP. 1 molecule of NADH
Yeah.
2.5 ATP. So as long as you know those relationships, you should be able to do any kind of math there.
So 1 molecule of each makes one. NADH makes 2.5 and FADH makes 2.
Yeah.
Cool.
I think the homework will help you with this. The homework is a lot of good practice on the concepts of these things.
Will we have like one question of this on our quiz like from this lecture today?
Yeah, the quiz has questions from chapters 18, 19, and 21.
Okay.
I don't specifically remember, I actually don't remember which quiz question I put on there, if it's regarding the material we covered today or yesterday, but both were chapter 21, so. Any other questions? Wasn't today cool? Wasn't this cool what we talked about? It's so cool. I hope you think it's cool the way I do. All right, well, that's it for today. So I will have my regular office hour. I'm actually gonna move it up just a touch because my daughter has a swim lesson at 4. So let's do 2:15 to 3:15. Okay, so instead of 2:30 to 3:30, I'll be on 2:15 to 3:15. Okay. All right, have a good one. I'll see you Monday. Bring your homework Monday. Bring your calculator Monday, Robin.
Quiz today?
Quiz today. It goes live at 11:30. You got 2 hours, but once you start it, you got 30 minutes. But you don't have to start it technically until 1 p.m. if you kind of want to spend some time looking over things, preparing.
Woohoo!
Woohoo! Guys, we only have 1 week left in this class after this.
1 week.
Well, 1 week of like new material.
I was
In
gonna say, don't we
my
have a
head,
whole
it's
week?
1 week because the week after is just review.
Yeah, I was like, we should have a whole week before.
We have 2 weeks technically, but we're only covering new material through the end of next week, possibly the Monday after. We'll see how next week goes. Next week we got a normal week. The week after we could have the exam Monday or Tuesday, depending on how next week goes. On the syllabus, it's set for Tuesday, but we'll see. Okay, have a good one.
Bye. Thank you.
Bye.
Have a good weekend.
You too.